I'm Ellie Juarez, and I just want to briefly tell you about the power of ultra-rich nanopore sequencing. And then I'll hand it off to really the star of the show here, Dr. Matt Luce, who will tell you about his tool, Robin, and how it brings a rich multi-omic view to CNS tumor profiling research studies. So very briefly, Oxford Nanopore is a new generation or third generation of DNA and RNA sequencing technology, and really the only technology that offers real-time analysis for rapid insights in a fully scalable format, from our MinION, which is pocket size, to our population scale instruments like the Promethean line. Our technology can analyze native, so no PCR copying required, DNA or RNA, and sequence at any length, both short to ultra long. ONT provides a convenient, easy to use, point and click end-to-end workflows for developed solutions, while simultaneously enabling researchers to develop new and exciting tools for ONT, like you'll hear from Dr. Matt Luce later in the talk. And all of this is enabled by a tiny nanopore. Embedded in a membrane, the DNA or RNA passes through this pore and emits a unique electrical signal that can be read. And these squiggles you see here on the video, each are slightly different. An A is different than a T and versus a G. And importantly, a methylated C is slightly different than a non-methylated C. And these unique properties of the nanopore sequencer provide both the genetic and epigenetic information from a sample, making ONT a perfect platform for multi-omic studies. And I think this comprehensive view is really crucial in the characterization of cancer, for which studies in the past 5 to 10 years have shown that that multi-omic comprehensive view of the genome is really crucial. So at the end of the day, cancer is a disease of the whole genome, which is characterized by both genetic and epigenetic changes. And these changes then drive both the initiation of cancer or oncogenesis and lead to mechanisms for evasion of cancer treatment and often, unfortunately, also to relapse. And so being able to get that multi-omic view and to probe SNVs with facing, epigenetic markers, fusion transcripts, et cetera, on a single platform is really what we think will be crucial for the community at large to continue to do the important work in studies to advance the oncology field forward. And so Dr. Luce's talk today will focus on how you can leverage that direct DNA sequencing to get both the epigenetic and genetic insight from the same sample simultaneously and in a single sequencing run. And so in this talk, he'll focus on 5MC for the rest of the webinar, but ONT sequencing can also provide other epigenetic markers like 5HMC, 6MA, as well as modifications of RNA like M6A, all while providing simultaneous detection of genetic variants like CNVs, SNPs, and structural variants, allowing you to face no additional prep or modifications required and no need for a separate sequencing run for data generation. So Matt will give you an amazing example of how nanopore sequencing can be leveraged for central nervous tumor profiling, but there are several studies and publications showing how ONT tumor methylation profiling can really revolutionize our understanding of other types of cancers like AML or pediatric cancers like retinoblastoma, where parent of origin is thought to play a critical role in long-term outcomes. And so all of this is through that direct RNA sequencing, rapid turnaround time and real-time results that don't require you to batch, but instead could enable you to action quickly with a single sample and providing that rich multi-omic view at an affordable price point. So without further ado, it's my pleasure to hand it over to Dr. Matt Luce, who has worked with nanopore data for over a decade, and he was involved in the sequencing of the human genome. His laboratory developed the first implementation of adaptive sampling, actually, and he's now applying these methods for rapid diagnostics of CNS tumors using Robin. So, Matt?
Thank you. So thanks for that introduction. It's a pleasure to be here presenting today on the work that we've been doing here in Nottingham on intraoperative CNS tumour classification. And in kicking off, as was already mentioned, we have been using nanopore sequencing for some time in Nottingham. I'm not sure if it goes back quite as far as Robin Hood, but I sort of think it should have done, and it was exactly what Robin was after in trying to share things with everyone and democratize access everywhere. Anyway, that's AI for you. Before I get going, I do need to say I'm not an employee of Oxford Nanopore Technologies, but I have received free flow cells and consumables for some of the experiments I'll discuss today. I'm not medically qualified, so I speak today from the perspective of a researcher and technical development. And all the work that I am showing here today is for research use only and hasn't been used in decision-making with respect to patient care. So I will talk at the end of this presentation a little bit about how hopefully that will start to change. So we've already had a bit of an overview of nanopore sequencing and how it works. This video shows a molecule being captured by a nanopore being sequenced. And one of the remarkable things that you can do with nanopore sequencing is you can actually work in real time with those data, and you can select the molecules that you want to sequence. So you can throw molecules away and you can choose to sequence another molecule. And you can do all of this in real time. And this adaptive sampling or read until, when we first started working on this many years ago, is one of those kind of space age ideas about sequencing that's been so exciting to be involved with. We were involved in establishing this method, and I just want to sort of draw a distinction between adaptive sampling and whole genome sequencing and explain some of the benefits, but also some of the things you don't get. So when you're doing whole genome sequencing, you expect to get uniform coverage of your reads across the whole genome that you're sequencing. But when you're running adaptive sampling, you're targeting a particular region of the genome for sequencing, and that target region is going to influence the data that you get. And essentially what you're doing is you're choosing to make those molecules that overlap with your target long, and the molecules which don't overlap with your target to be short. What does that do? Well, it allows you to accrue coverage over key genes. So I'm showing here an animation of coverage accruing over BRCA1, which would be a target in a cancer panel. And so you get more coverage faster. In addition to that, all those other reads that you're not sequencing to full length provide you with information across the genome. They provide you with information on copy number and copy number changes. And here I'm showing a tumour sample, and you can really clearly see those copy number changes as a consequence of binning the read data. Because Nanopore gives you multiple sources of data, you also get methylation. Here I'm illustrating methylation coverage over a specific promoter in the genome using methylartist to visualize that coverage. But you can look at methylation genome-wide in an adaptive sampling experiment by considering the totality of the data. You can also look for things like gene fusions, and you can look for point mutations if you have sufficient data. So we get multiple pieces of information from a single experiment. And critically, we get this information in real time. So this is an animated GIF that I've been showing for a few years now. This is showing 3 different samples that we've barcoded on a single sequencing run. And we're binning the data and we're plotting the copy number changes. And in this case, we were doing it every hour. So each one of these frames corresponds to 1 hour of sequencing. And if you watch really carefully, you can see very quickly, of course, those copy number changes emerge. So we can get rich data from this single experiment type. So we developed these methods initially back in 2016, and then we released updated methods in 2020, which allowed us to do this at the scale of human genome. And those rich methods now have been incorporated into Nanopore's sequencing platform. And so you can get all of these things: single nucleotide variants over genes of interest, specific gene fusion events, promoter methylation, copy number changes, and global methylation. And when we published this in 2020, we were very excited about our findings, of course. But something else happened in 2020 which diverted the world away from thinking about this, and we spent some time looking at small viruses. But actually, I was fortunate enough to be contacted by a group from Heidelberg. Felix Sam and colleagues, and in particular his then PhD student, Ariba Patel, got in touch. And sadly, I wasn't able to visit Heidelberg then. I have visited since. It's lovely, and I strongly recommend it. But they got in touch with me because they saw the paper and they had a very specific problem that they felt that this would apply very well to. So the problem, of course, was CNS tumours. And if you're not aware, the stats I'll present here are UK specific, but in the UK, brain tumours are the leading cause of cancer death in people under the age of 40. And there's over 12,000 new diagnoses each year. And sadly, only 15% of people who have the most common brain tumour types are alive 5 years after diagnosis. And so this means that accurate, timely classification of the tumours, diagnosis, is absolutely critical, both to treatment and research. The quicker you can get patients onto the right pathway, the better. So this is a particular challenge, and of course the enormous teams that work together to do this have to work together in a coordinated way. CNS tumour diagnostics are challenging and rely on integrating multiple different data types and pooling that information together to give a classification of a specific tumour. And so this is a challenging process that takes plenty of time and effort. Why is it important to get this right? Well, this is an indicative survival curve for 3 different groups of patients. And of course, you want to be able to tell the patient which group they are likely to be in. You want to be able to assign them on some basis. And it turns out that this assignment is based on very simple differences, in this case, between these particular tumours. So tumours that look same can behave very differently. So here we have a subset of tumours that have been stratified by whether the MGMT promoter is methylated and then whether or not they have got homozygous deletion of the CDKN2A gene. And just by measuring those 2 things, you can work out a prognosis for patients. But of course, to do that requires at least 2 tests currently, one to look at the methylation status of the promoter and another to look at the possibility of gene deletion. Now, this is another reason that we have to look at tumours quite carefully to work out our classifications. Here we have sections through 2 tumours that look very, very different. I am not a histopathologist. It's not my area of expertise. I would not classify these tumours as being the same if I was looking at the images alone. If you look, however, at the copy number profiles that we're showing below, there is some commonality here. Gain of 7, loss of 10, absolutely characteristic of the specific tumour type. So just looking at the histopathology may not be sufficient to allow you to classify 2 tumours. And then we have an example here where we have a tumour entity that just simply doesn't have a clear defining morphological feature. And this is an example of something which pathologists classified as being something called a malignant ectomesencymoma, based on all the available data that they had. But there was something new on the horizon that could be done at this point. And what was that? Well, this was the fantastic work of David Kapper and colleagues published in 2018, which actually showed that tumours could be classified on the basis of their methylation profiles. So just by looking at the changes in methylation status of the tumour tissue, you can see very clearly on the right-hand side here the clustering of these different tumour types. And so by looking at the methylation across the genome, you can assign a tumour to a specific classification. And just thinking back to that previous sample that we looked at, that previous sample, when analysed based on the clinical data alone, that diagnosis was incorrect. This is a different tumour type based on the methylation, and it actually has a characteristic fusion that subsequently could be identified. So there's plenty of reasons why it's challenging to classify distinct tumour types. So what if we could combine all of these different tests together into a single assay? I've given you examples there of copy number changes, of presence or absence of fusions, methylation classification. What if we could do all of this in one assay? And it was that question which the group in Heidelberg were interested in trying to get to the bottom of. And so they proposed that we should be able to do this from nanopore data alone. And we used a targeting approach, an adaptive sampling approach, to allow us to look at specific genes in more detail and to come up with this accelerated process. And what we found was, yes, in 3 days in this case, using a gridiron, it was possible to get a complete molecular classification of a tumour addressing all of these specific questions about it in a single assay. So in contrast with the current approach where you would have to collect your sample, extract DNA, one portion of that sample would go for standard sequencing, another portion would go for bisulfite conversion and array analysis. Everything could be done in one assay on a nanopore sequencing device. Now here we show the timeline for these as both being around 5 days. In reality, what we're missing here is that the conventional pathway requires you to gather enough samples to run through your analysis pipeline and your library and laboratory pipeline, whereas with nanopore sequencing, you can just start sequencing on the device once you've extracted your DNA. So we are very pleased to be involved in this paper and work with Ariba and colleagues on doing this, and we showed it was possible to do this with nanopore. And this is an example of the paper when it came out in 2022, where we showed the ability to get both diagnostic, prognostic, and therapeutic targetable mutations potentially from this single assay. Having done this and published it, it was fabulous to start meeting new people. And one of the people I met was the gentleman in the bottom right here, Simon Payne, a neuropathologist here in Nottingham. And Simon had seen this paper and came to see me and told me this story of a patient that he had seen in 2017, an 11-year-old girl who presented with seizures. He had an intracerebral tumour, and the radiologists were unsure as to what it was, what the grade of the tumour was. And so they carried out further analysis and came up with this diagnosis of a gliosarcoma variant of glioblastoma, which is a grade 4 tumour. But there was something about this diagnosis that didn't sit comfortably with Simon. And so at the time, this is 2017, this was before the array paper had been published, but obviously the community was well aware of what the methylation data could tell us. And so he sent that sample for methylation array analysis. And the conclusion of that was actually that this patient did not have the glioblastoma type that was suspected, but actually had a schwannoma. An intracerebral schwannoma that's a low-grade tumour that was surgically treatable and, in fact, had been surgically treated as a consequence of the removal of the tumour. And 7 years later, there was no evidence of recurrence in that patient. Obviously, that's fabulous for the patient, but critically, it meant that unnecessary radiotherapy had been avoided, but only just. And so Simon's question was, can we try nanopore sequencing here in Nottingham? And could we use it to try and speed up the turnaround times for the patients that he saw day in and day out? Why is that important? Well, obviously, every country has its own methods and approaches and its own setups for doing this sort of thing. And I can only comment on what we have in the UK. In the UK, there are distributed centres that can provide various aspects of the testing, but typically they are not where we are. So in Nottingham, we don't have a local array facility, and so samples are sent to London. If you look at the timing of that, it takes time to collect the sample, to prepare it for shipment, to then send it and have it received. Then those samples will need to be batched up to be run effectively on the array. That can take some time. Running the array itself will take another few days, and then reporting has to come back to the clinicians. In total, you're looking at a significant delay in obtaining just the methylation classification. And of course, this is incredibly important for clinicians that you can't get a fast diagnosis. It's possible that the grade of the diagnosis may change. It means that MDTs, the multidisciplinary team meetings where clinicians discuss patients, can often just be waiting for a result. The correct approach with respect to treatment, so oncology, post-surgery, and even surgical decisions themselves may have been better informed had you had knowledge of a specific classification or confirmation of a classification. Those are just thinking about the clinicians. We're not thinking about the patients who have to deal with the delay in knowing, and their families in knowing, the outcome. And I mentioned here let's not even mention whole genome sequencing and panels and such, because they will typically be ordered after you've got the array. So the delays build and build. So as a consequence of all of this, we were very grateful to receive some initial funding, actually from the University Hospital Trust themselves, to allow us to evaluate 30 cases retrospectively here in Nottingham. And we compared these cases against the nanopore classifications we obtained and with the actual known diagnoses. And you can see here there are good concordance with the final diagnosis, 80% of the samples. There's then a subset of samples that were more complex. In all of those cases, we could resolve those differences. It could be tissue sampling, because for the retrospective cases, we weren't going from exactly the same material that had been sent for diagnosis. In some cases, it was differences in classifier versions, and in some cases, it was a completely new entity that was simply not in the classifier. But based on this, these initial 30 cases, we were very happy with the results and wanted to proceed further. Now, I don't want to, for a moment, suggest that we're the only group who are doing this. There are many groups around the world. I've tried to reference the various papers that have been published since 2017 looking at this type of work. They have all used slightly different approaches. They go from different tissue types in terms of being frozen or fixed. They use perhaps different library methods. Some of them are faster or slower, and different people have looked at different things. Amongst these approaches, it was only the one from Ariba and ourselves that actually looked at all of these different aspects. So they gave a classification, they looked at copy number variation, they looked at structural variants, single nucleotide variants, and MGMT promoter methylation in a single assay. The challenge was that still was taking days to get a result. And so we wondered whether we could get this any faster. We wondered if we could bring those days down to hours, because if we could do that, we could start influencing the surgical process, but also give results back to patients far faster. So to do this, we set ourselves the goal of coming up with a diagnosis, or sorry, a final classification, final molecular data within 24 hours, and methylation classification within 2 hours of receipt of sample from theatre. And so this cartoon shows our workflow. Sample is received, DNA is extracted, takes around 30 minutes. We're using the standard DNA extraction protocols from the hospital. Then we have a library preparation step, and that takes us around 60 minutes. Now, currently we are using a modified protocol based on Nanopore's ultra-long sequencing kit. We're not getting ultra-long reads, we're getting normal read lengths, but we find that this approach works very well. That's because the alternative methods, which is a rapid prep, which is incredibly fast, 10 minutes, but doesn't perhaps give you the longevity of sequencing that you want. So we wanted to collect a lot of data over 24 hours, or a ligation approach that at that point took 2 hours. That was too slow for us to meet our target, so we developed this newer protocol. So we start sequencing typically within 90 minutes of the sample being delivered to the pathology lab. And within 2 hours, sometimes even quicker, we have our initial classifications. We keep sequencing then for 24 hours, and we have our final molecular data available the next day. In order to get that throughput, we've switched from using MinION to using Promethean. And in this case, we're using the P2i. We've also used the P2 Solo and the Promethean 24 to get similar data. We wanted a particular N50 for our reads, because if you're using adaptive sampling, you want that to be not too long, not too short. We were aiming for around 10 to 12 kb, and that's exactly where our protocol ended up. I'm showing here pictures of Simon Deacon and Swasti Kiani, who were the team who optimized this protocol and made it work well for us. And you can see the read length distributions and the sort of coverage that we were able to get in 24 hours. I would say with further refinement, we're able to get the Promethean coverage in 24 hours closer to 40 to 50x. And Nanopore have recently provided to us a faster ligation protocol, which we're just testing, but we're very encouraged by the results we're seeing there as well. That also has a similar kind of 1 hour library prep time. So we think we can make this fast. Problem is we needed to analyze all of these data, and so this is where Robin comes in, and people ask, well, what is Robin? Robin stands for Rapid Nanopore Brain IntraoperatIve Classification. My colleague Simon Payne is responsible for the dreadful backronym that we have here, but we have developed this tool, and it exploits the ability of nanopore sequencing data to be analyzed in real time as they come off the sequencer. So this is a web-based tool. We don't run it in the cloud, we run it locally on device. So this will run on the P2i or the P24. It will run on the compute that you have associated with your P2 Solo, and it will generate all of those outputs, the methylation classifications, the copy number changes, the coverage information you need. It will run SNP calling if you have sufficient data available for SNP calling to be run. It will predict gene fusions and give you MGMT promoter methylation. And we're developing this, and it's available at the GitHub site shown here. So we have now run this on 100 cases, going from diagnostic, so going from tissue, from surgery, and taking this straight through. Our concordance has improved. We think that it's improved for several reasons, but one is tissue selection is critical, and we're going from fresh tissue, so we're up to 84% concordance. We have some novel entities. We have some samples that it's classifier version specific, whether we'll detect them, and only a minority have struggled from a low tumour fraction. There are some which are unclassifiable both by standard of care and Nanopore. So we're very happy with what we're seeing. And I wanted to just share with you a few of those stories that we've seen to illustrate how this helps. So this is a case that we saw very early on. It was the second or third case, a ZFTA-fused supratentorial ependymoma. And this case was an 11-month-old child, and the intraoperative smear opinion was that this was a primitive tumour with a wide differential diagnosis. And the surgical decision was to have a subtotal resection and await definitive diagnosis. what happened subsequently was that this was confirmed to be an ependymoma, and a second surgery was needed for this child. Now, this was the first time we had been running Robin alongside alongside a surgery. So in that intraoperative timeline, where I let the pathologists know, they didn't act on this, but we contacted them. And this and video, if it works, is going to show you what we were seeing at that time. So we've got a few plots here that will generate, and this is showing you that within minutes of sequencing starting, we're using two different classifiers here. Within minutes of sequencing starting, both of our classifiers were telling us that this was an ependymoma. And I contacted the pathologists and said, you know, this is what we're seeing. And they're 4 floors down from me in our medical school. And within about 2 minutes, there were 3 pathologists at my door wanting to see the data. And this was confirmed very, very, very quickly. And we knew that information. We couldn't pass it on, research use only. We couldn't pass it on at that time to the surgeons. But we had the classification, and not only did we have the classification, but the next morning we had the evidence. This is a plot taken from Robin, which shows reads labeled by color and precisely determines the position of the breakpoint and the evidence for that gene fusion that's predicted for that tumor type. So in theory, we would have been able to save a second surgery. This is another child case, in this case, who presented a 3-year-old, short history of vomiting, a posterior fossa tumour, and the intraoperative smear, ATRT was favoured, but medulloblastoma was possible. Now, in this case, we started sequencing, and we very quickly had a classification, and now I'm showing 3 different classifiers that we use, which confirm this to be medulloblastoma. Almost immediately, the 3 classifiers were in agreement. Not only did we have that information, we also had information on copy number variation, and in fact, we could show by looking at that the MYC gene was amplified as well. So this diagnosis is obviously significant for the patient. But I just want to show you, because we track this information, how long it took to get this information by standard of care. It took 44 days for all of that information to be confirmed. Multiple meetings of multiple clinicians, unable to discuss in detail the case, but 44 days. Had we been able to use our research use only Robin timeline for this, Day 0 was the operation. On day 0, we had the complete classification, the medulloblastoma, group 3, CNS, grade 4 classification, the confident classification from all of 3 classifiers. And the following day, we had knowledge of the MYC amplification, and that could have been presented to the MDT, which, as it happened, was on the second day relative to the operation. Had we been able to do that, we would have been able to get that result 43 days, give or take, before the standard of care timeline. So this can be absolutely transformative for these patients. The next case study I just want to briefly present is that of an 18-year-old male. This individual came into the hospital after a head injury. They had a CT scan and identified an incidental lesion. There were various question marks over what tumour type this was. Was it a high-grade glioma? Was it a metastasis? There was no sign of tumour anywhere else. And so we had this sample, and here I am showing 3 plots. These 3 plots identified this unbelievably to be a schwannoma, that very same tumour case type that had got Simon interested in using Nanopore originally. But not only that, Simon said if this really is this schwannoma type, it should have a particular fusion. And so we were able to go and look and identify exactly that fusion in our data, all within the 24-hour time window that we had set ourselves. So yes, very rare tumour types, but we get that additional wealth of information that we are after. So I've shown you that actually it is possible to do this, and we can run these classifications very easily, local to the patients. This was the correct diagnosis, and in this case, various reasons, the standard of care was even slower than we would like, but it was confirmed to be the correct diagnosis. So what's unique about our approach? Well, firstly, I should be clear, it builds on so much work from so many other groups. And we are so grateful to them for sharing everything that they have done. It implements that real-time nature of nanopore sequencing. It enables us using our graphical interface to see what's happening in real time. We actually use multiple classifiers rather than relying on a single classifier. The reason we do that is that we found that different classifiers have different properties. They're a little bit like having one or two pathologists and asking them to come to agreement on what they are seeing. We enable people to see the classification dynamics. I've not shown any examples of this, but it can be quite useful if you've got some unusual performing sample to have a look back and see exactly what has happened over the last few minutes of sequencing. Obviously, because we have switched to Promethean from MinION, we're getting more data. And so we get that those extra amounts of data that let us see more in the same amount of time. We've got a very fast library prep, which we're very happy with. It gives us exactly the read length distribution that we want. But having said that, we are also trying this ligation protocol, and in our view, you will pick the right protocol to use for the specific experiment type that you are after. So if you're after a very fast intraoperative classification, you may go that way. If you're after results within 24 hours, you might go for a ligation approach. And by using the panel sequencing, the adaptive sampling, we're able to get exactly the information we need on key genes and events that might be typical of a specific tumour type. So what does this mean? Well, one of the things that we see increasingly is, of course, depending on where you are in a geographic area, you may be close to or far away from an area which is able to offer the sort of diagnosis that you need. The ability to take something like a P2 solar or a P2 ion, put it anywhere, put it close to patients, will give us complete equity of access. Everybody will be able to have the same type of treatment. It will give us speed, it will give us accuracy and efficiency. You won't have to wait for those results. All the work transferring samples, it can all be done next to the patients. It means that you'll get results when the patient needs them. So for those cases where really knowing what's happening intraoperatively is important, that's possible, and we call that Ultrafast. And if we need it for the next multidisciplinary team meeting, then that's rapid. We're still talking the order of hours. There are some very interesting possibilities opening up in terms of where you do your analysis and how you run your testing. If you're sequencing close to patients, you don't necessarily have to do the analysis close to patients. The sequencing can be done, but then the data are very transferable and can be shared and used for analysis. One of the things that we have is multiple sites starting to use this, and I think it will be obviously highly important to use cross-site validation to share best practice and to arrange things like sample swaps to make sure everyone's working effectively and efficiently. And I think that this is complementary to what's already happening. In the UK, we have whole genome sequencing, we have Genomics England driving a huge number of samples through, and that's absolutely fabulous. This allows us to almost sort of uncouple the rapid classification that's required from the longer term discovery process that happens through whole genome sequencing. But having told you about Robin, I just want to mention Batman, which is something that we are working on. And this is a unique feature of adaptive sampling. Batman is breakpoint adaptive targeting alongside methylation analysis on Nanopore. What does Batman allow us to do? Well, I'm showing you here a copy number plot from covering the locus of CDKN2A2B. And you can see clearly the change there in coverage that's indicative of a deletion. What we can do is we can analyze these data whilst we are sequencing to predict exactly the position of that breakpoint. And you can see those predictions being made in the panel below. And by doing that, because we have the ability to change the targets that the Nanopore sequencer is looking at, we can actually get it to target those specific sites. And when it targets those specific sites, we can uncover reads that confirm precisely the deletion. And we've started running this now on some of our research use only samples. CDKN2AB deletion is a relatively straightforward approach and place to use this, but we can actually start to distinguish precisely the events that are happening. So we're quite excited about what this may allow us to do in real time, but I hasten to add that this absolutely is research use only at present. So rapid diagnostic information, rapid classification information is possible. The photograph here is from a patient sample analysis room that is adjacent to the theatres in Nottingham, the surgical theatres. And you can just about see on the desk there, the sequencer that we used. Effectively, this is a storage cupboard we set up next to the theatres to try and make this as fast as possible. Intraoperative use is readily achievable, but there are advantages to using multiple classifiers, and there are advantages to using this for clinicians, for patients, and it's going to be really exciting to see what happens. We do have a preprint, which is up on medRxiv. Please do have a look at that. It's actually only describing the first 50 cases that we looked at. We've generated a lot more data since then. And I would like to acknowledge gratefully the support I've had from the NHS in Nottingham, Dr. Simon Deacon on the right here in the photo, PhD student who has done all of the work developing this, Mr. Stuart Smith, who is the surgeon who's been so important to driving this forward, Rob Goldspring, Dan Martin and Amanda Corkill, who all work within the NHS and have facilitated us getting this on board. And of course, our collaborators in Heidelberg and Sebastian Brandner in London, who've been incredibly helpful working this through with us. So thank you very much for listening.